Hi, my name is Saoirse Connolly, and I'm going to be talking briefly about my project involving viral RNA detection using a fluorogenic RNA-based CRISPR-Cas13a system. Nucleic acid detection was seen to be instrumental in the mass viral disease and diagnostic testing during the coronavirus pandemic. Sensitive and accurate nucleic acid detection will remain relevant in the future as novel viral diseases emerge, as well as novel strains of existing viruses. The creation of a detection platform that is both sensitive and accurate can be achieved by harnessing the adaptive immune system of bacteria via clustered, regularly interspaced, short palindromic repeat sequences, or CRISPR, as well as the Cas13a nuclease. When invaded by foreign genetic elements, bacteria utilize adaptive immunity to prevent future infections via CRISPR-Cas systems. When the invader is identified, the CRISPR-Cas system directs nucleases to bind and cut specific sequences of the foreign genetic material. These small segments get incorporated into the genome of the host bacteria as CRISPR arrays. And it is the transcription of these CRISPR arrays that lead to the creation of CRRNA that can bind to Cas nucleases. The binding of CRRNA to Cas provides the specificity of the nuclease via complementary base pairing of the complex with target nucleic acids. The binding of Cas proteins to their guide is highly specific, and the programmability makes them desirable for nucleic acid detection. The Cas13a locus contains two nuclease domains termed HEPN domains. Upon recognition of a single strand of RNA via complementary base pairing with the CRRNA, these nuclease domains activate and transcleavage activity occurs. This means that activation of the Cas13a protein leads to random promiscuous cleavage of nearby RNA. In terms of this specific detection system, this will lead to the cleavage of the fluorogenic broccoli reporter. A cleaved broccoli reporter can no longer properly interact with the DFHBI and therefore can no longer emit the same intensity of fluorescence. So, in the presence of an active Cas13a, broccoli fluorescence will decrease. Initially, PCR was performed to obtain the DNA templates that would be used for T7 transcription. After T7 transcription was performed, a 10% denaturing gel electrophoresis was used to determine if the correct transcripts were obtained. The results shown here determined that the transcription was successful based on the length of each RNA transcript. Upon binding to DFHBI1T, a folded broccoli emits green fluorescence. So, an initial experiment was performed to determine if the RNA transcripts would alter the binding selectivity of broccoli to DFHBI1T. Each transcript was incubated with DFHBI for a total of 90 minutes at room temperature. The fluorescence was recorded at 503 nanometers after every 30 minutes. It was found that the target RNA in the CRRNA did not interact with the DFHBI and emit fluorescence at any time interval. It was also observed that after a prolonged time, the fluorescence intensity of broccoli decreased. This is hypothesized to be due to the degradation of the broccoli RNA afterward. If the broccoli is getting degraded, it is no longer able to properly interact with DFHBI and emit fluorescence. Next, the fluorescence of broccoli was measured in the presence of Cas13a to determine if the nuclease could cleave the reporter after all. Before activation, it can be seen that the protein does not have an effect on broccoli fluorescence. However, after the complex is activated through the binding of CRRNA and the target RNA, there is a decrease in the measured fluorescence intensity of broccoli at 503 nanometers after just 30 minutes at room temperature. The change in fluorescence intensity of the broccoli reporter was increased after the activated CRISPR-Cas13a system and the positive control were incubated for 20 more minutes at 37 degrees Celsius. The longer incubation time led to a marked decrease in fluorescence intensity due to the inability of unfolded broccoli aptamers to interact with the DFHBI after the promiscuous transcleavage activity of the active Cas nuclease. The fluorescence intensity of broccoli was then measured in a new reaction buffer to determine if the buffer could enhance the activity of the CRISPR-Cas13a complex. All previous experimentation occurred in a broccoli folding buffer, so the activity of this buffer was compared to a Cas13 reaction buffer. The broccoli folding buffer allows for broccoli to fully fold and interact with the DFHBI to emit fluorescence. The broccoli folding buffer does not interfere with the activity of the CRISPR-Cas13a complex because when activated by the recognition and binding of its target, there is a measurable decrease in fluorescence. So the nuclease activity is unaffected. The Cas13 reaction buffer affects the initial folding of broccoli, making the interaction between broccoli and DFHBI less favorable, leading to a lowered initial fluorescence even in the absence of the active CRISPR-Cas13a complex. In the presence of the active complex, there is a slight decrease in fluorescence observed. Still, however, the decrease is much less than in the broccoli folding buffer. The broccoli folding buffer allows for the broccoli aptamer to interact with its target as well as for the complex to fully activate. Despite allowing nuclease activity, the Cas13 reaction buffer does not allow for the broccoli aptamer to fully fold and function properly. Therefore, it is not the best buffer choice for future experimentation and optimization for an accurate system. In conclusion, viral RNA detection using a fluorogenic RNA-based CRISPR-Cas13a system is possible. It was first seen that DFHBI does not emit fluorescence on its own or interact with the CRRNA or target RNA to emit fluorescence. This means that in this system, fluorescence is only occurring when the folded broccoli aptamer is interacting with DFHBI, and it is the sole reporter of fluorescence. Incubation times can negatively affect the system, as longer incubation could be correlated with a decreased fluorescence due to the degradation of broccoli RNA and non-active Cas protein. This system performs best in the broccoli folding buffer compared to the Cas13 reaction buffer because the broccoli folding buffer allows the reporter to accurately fold and interact with DFHBI, as well as for the active CRISPR-Cas13 system to cleave these reporters. In the future, the concentration of the Cas13a protein will be assessed in order to determine if a change in protein concentration will lead to a change in broccoli fluorescence. A future goal is to have a highly sensitive and accurate platform with as little protein and RNA concentration as possible. The sensitivity of the platform will be assessed in the future by determining the minimum concentration of target viral RNA the system can accurately detect. Furthermore, the system's accuracy will be determined through the incubation with RNA that differs from the target RNA by only a few mismatched bases. This is vital in order to detect between differing strains of viral RNA in the future. The major end goal of this project is utilization in a clinical disease diagnostic setting. Once the system has been optimized to become as accurate and sensitive as possible, it will serve as a point-of-care option for viral detection. I would like to acknowledge and thank Dr. Mingzhu Yu, Qian Tian, and the rest of the U Lab for all of their advice and input during the experimentation with this project.